Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in Immunology we will be talking about regulation of immune response part 1. To discuss this topic we have with us our subject expert Dr. Anita Kamra Verma. Dr. Verma is an Associate Professor in Department of Zoology in Kirori College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Good evening everybody. I am Dr. Anita and thank you for the introduction Amrit. And today I am going to speak on another topic in immunology, that is regulation of immune responses. Now regulation itself means controlling, and immune responses first mean that what exactly are the responses, immunological responses of the body. So I am going to start with immunoregulation first. What is immunoregulation? It is a balance between activation, suppression of effector cells to achieve an efficient immune response without harming the host. That means there has to be activation, which in normal conditions we call it as the body's immunity, and we have suppression. That means we are tolerating the infection and we are immunodeficient. That means there is a condition of immunodeficiency. And when there is activation, it might also be autoimmunity. Now, why do we want to understand this whole concept is the significance is that the tolerance may have potential importance in graft versus host reaction. That means I want to treat an autoimmune disease or I want to treat some allergies, and we can also prevent certain immune responses which would be highlighted during a gene therapy. All these sound very big, but let us start from the basics. What are the key players of the immune system? Let me take you back to your initial classes which you have done. We have white blood cells, which are the leukocytes. Leukocytes are basically polymorphonuclear granulocytes. That means they consist of neutrophils, basophils and eosinophils. Then we have the specialized immune cells, which are the lymphocytes, which are the T cells. T cells are helper cells and cytotoxic T cells. We have B cells and we have natural killer cells. Also, we have monocytes, that is macrophages, and dendritic cells. All these you have heard of. So, I will divide the lecture briefly into the cells of the innate immune system, because this is the innate immune response which it will be responsible for, and we will have cells of the adaptive immune system. So quickly, if we breeze through, it is macrophages will belong to the innate immune system. They are large and they are particles or cells which can actually engulf the particles. What is their function? It is to ingest and eliminate the infectious agents and any processes or parts of a particular molecule which is presented as an antigen. In my last class also I had explained what is an antigen. Antigen is anything which is foreign to the body. It could be live or it could be dead. It could be a pathogen. All antigens are not immunogens. Immunogens will be only those molecules which are going to elicit an immune response. And what do macrophages do? Independently, they can engulf and eliminate particles, but other than that, they can also regulate other immune cells. That means my macrophages will belong to both innate as well as adaptive. Now, this is a cartoon which shows you how the macrophages are capable of giving out pseudopodia. Pseudopodia are nothing but false feet. They can actually go up to the bacteria, recognize them, engulf them, and they can put them in. You can see these are the vesicles in which they have kept the bacteria. These are the undigested bacteria, and you can see that the entire cell has different pockets. These are small pseudopodias and these are the long protoplasmic processes of the pseudopodia. Now, this is an entire cell. It is going to digest the bacteria. This is the Golgi complex. This will be the endoplasmic reticulum. So, when I look at it, this macrophage will help you remember all what you have studied earlier. Moving on, what are the second type of cell which belongs to the innate system are dendritic cells. What are dendritic cells? That also I had revised with you last time, that they are cells which have large processes and they are long cytoplasmic processes. They appear as dendrites. Where do they live? The dendritic cells are present in the tissues in immature forms, and they will only mature when they are subjected to an infection or there is an injury of any part of the tissue, whereby they elicit an innate response. Can the mature dendritic cells travel from the tissues? Yes, they can travel through the lymphoid organs, that is lymph nodes and spleen, but they can attach the antigen on their surface. So, they can actually move with that, and that is how they work as antigen presenting cells. That means dendritic cells are capable of recognizing the antigen, making a complex with the antigen and then putting them as antigen presenting cells. They can also influence the activated T cells. Next type are the natural killer cells. Natural killer cells are basically just present in the background, and they are capable of killing non-specifically, but they kill the target cells specifically. That means they do not arise because of a certain infection, but they are constitutively present to eliminate and target the cells. They are large granular lymphocytes, and they do not need to be primed. Primed is that do not have to be shown the infection to get activated. Dendritic cells and macrophages require priming. That means they need to recognize the infectious agent to get activated. Then the second is the cells of the adaptive immune responses or the immune system. T cells, we have already gone through T cells. These cells are mature cells of the thymus. That means what does a T cell do in the thymus? It learns how to differentiate between self and non-self. If our cells are not capable of recognizing the cells, they will cause autoimmune disorder in the system. There are two main types of T cells, T helper cells and cytotoxic T cells. That is, T cells which are recognized or which are expressing CD4 are the T helper cells, and the ones which are recognizing the T cells, the CD8 receptors, they are called as the cytotoxic T cells. They are responsible for specific recognition of antigens, and they can retain memory of an antigen. This is an important part. The next type of cells which are responsible are the B cells. B cells, unlike the T cells, they mature in the bone marrow, and they are called B cells because initially B cells were known to be maturing in the bursa fabricus, which is present in the cloaca of the birds. That is how it got the designation B cells. B cells are capable of recognizing the antigen in soluble form, and they can produce antibodies. So, B cell can actually produce soluble mediators which are antibodies in the solution, or they can attach the antibodies on the surface of the B cells. The B cells have the capability of maturing into plasma cells and memory cells. This is also important because the infection can cause a memory within the system, and the memory is responsible only because of the antibodies which are circulating. Now, what have I understood till now? We have understood that there are three defining characteristics of the immune system. That means T cells are responsible for recognition or discrimination between self and non-self, that is host and non-host. it has memory. So that means once the antigen has been encountered, it will allow the immune system to react faster when another infection comes in. And this is the very basis of the entire system of vaccination. Also, there is specificity because the response is only to the pathogen which is present at that particular time in the host. So the specificity, recognition of self and non-self, and memory. These are the three characteristics of the immune system which we must remember to finally have controls on it. Now, the first line of immune defense was what? The neutrophils and the macrophages. That means the moment an infection comes into the system, who will rush? The macrophages will rush. They will display their antigen on the surface and they will signal other cells to get activated. That means I can activate just by putting the antigens and showing it to the T cells, or I can actually produce another soluble molecule which is an interleukin 1, which will also promote cell activity. The natural killer cells, what were they doing? I explained what are natural killer cells, but what will they do? They will also go and try and kill the infected cells or the tumor cell which is different to the normal cell, and it will release all the toxic substances into the cells. Where are the natural killers primed? I told you natural killer cells do not require priming, but they are trained in the tonsils, spleen or lymph nodes to do the function what they need to do. What else can natural killer cells do? Natural killer cells can produce interferon gamma and they can enhance the killing of viral infections, because the viruses have this ability to infect the cells and give out interferon gamma. So interferon gamma is a marker for a viral infection. If we use experiments in vitro, we see that the natural killer cells are capable of killing a tumor cell within 3 hours. So that means, can I use this natural killer cells activity from in vivo in an in vitro experiment? Yes, we can do that too. So my first line of defense was natural killer cells, macrophages. So what is my second line of defense? My second line of defense requires specificity. That means now the immune system has to have an antigen and a receptor where it goes and binds, and then it will form a complex which will activate the T cells, and by giving out some toxins or by giving out some cytokines. Also, T helper cells are incapable of recognizing the antigen as in its soluble form. It can only recognize the antigen when it is in a complex. That means it has to be presented either by the B cell or by the antigen presenting cell. So T helper cells themselves cannot kill like the macrophages or the natural killer cells. T helper cell, as the name denotes, can only enhance the function of other cells. That means it can activate the T cells by releasing cytokines. It can also help the HIV to further on kill the helper cells. That means I had also mentioned about antigen presenting cells in my last class. What were the antigen presenting cells? The dendritic cells, the macrophages and the B cells. What do they do? Again, this small cartoon will help you recognize and remember. Macrophages can phagocytose, parts of the bacteria can be processed, and the antigen processing will express the molecules like the MHC class 1 and MHC class 2 for T helper cell as well as the TC cells. So we are basically presenting the antigen to the T cell to get it activated. So here what do we see? We see the activation of the T cells. How? Not by recognizing the antigen, but recognizing the processed antigen which is being expressed here. So whatever I had taught you last time is again summed up in this small cartoon. Next, I want to talk about the B cells. Now, B cell also has very specific targets. That means, I am sure you must have heard about humoral immunity and cell-mediated immunity. It is very important to understand all the terms before we talk about the regulation. So, when I talk of humoral immunity, it is only means an immunity caused by soluble mediators. What are the soluble mediators? The antibodies. Who secretes the antibodies? The B cells. So, that means these antibodies are also called as immunoglobulins. And these immunoglobulins have 5 subsets. I am sure you must have done it in your previous classes. What do these immunoglobulins do? These antibodies, they help to activate other cells as well as kill other cells. That means, are the antibodies capable of killing the antigens? No. They are only capable of neutralizing the antigens. Antibodies are capable of activating T cells when they are in complex with other cells. Can they facilitate natural killer cells? Yes. Can they facilitate T cells? Yes. They both can facilitate them to further kill, but they themselves, antibodies themselves, cannot kill. So, this cartoon again for you to revise. What does the B cell do? This is capable of recognizing the antigen. When it recognizes the antigen, it can either express the antibody on the surface like it has done here, or it can release the antibody as soluble mediator into the blood. Once the antibody or the antigen make a complex, they can move to the helper cell, which is going to further differentiate into plasma cell and memory cell. This is for the proliferation of B cells. That means, once the B cell recognize a specific type of an antigen, there will be a clonal population, there will be increase of the number of B cells, so that they are capable of releasing more and more antibodies into the system. Out of these cells, some of the cells will mature into memory cells, and once the memory cells are made, they will remain in the system till they have another infection where the response will be much higher, because they would be recognizing the antigen before. What happens to plasma cells? Plasma cells go on releasing the antibodies, and these antibodies are released as soluble mediators into the system. Sorry. Now, when you go back, you will see that the antibodies are forming a complex with the antigen, and with the help of this, the macrophages are able to recognize it much further. That means, there are 2 things which happens. There can be covering of the antigen with the help of serum proteins. It is called as opsonization, which I have not marked here, but the antigen antibody complex is recognized by the macrophage itself, and once it goes into the memory cell, it remains as a red alert that the cells and the body's immune system is forever ready for another infection. So, what are the important features of the immune regulation? What we have understood till now, that there is specificity of the antigen. They can recognize a specific antigen, but I would rather say a certain part of the antigen, that is called as the epitope. It can affect T cells as well as B cells. Then there is another concept of tolerance versus activation. Now, tolerance versus activation is one other thing which I had said in my first slide, which is the concept which has to be discussed today on the base of whatever I am explaining. That means, I need to understand what is the antigen, what is the nature of the antigen, how is there a response to the antigen. That means, when, where, how was the antigen encountered, how much of the antigen was encountered. That will lead to an immune response. So, the magnitude of the immune response can be driven by the antigens that are capable of activating the lymphocytes, or the negative regulatory influences which will prevent or dampen the response. the other part which is important is the regulatory mechanisms which will act on all phases of the immune response. That means recognition, activation, as well as effector function. So, I will talk about this in 3 phases. That means I will talk about the regulation by the antigen, regulation by the antibody, regulation by the cytokines, regulation by the neuroendocrine function, or regulation by genetic factors. So, to begin with, I will start with the 3 first. That means regulation by antigen. So, what does the antigen have to do? The antigen needs to be recognized. So, what is important is recognition followed by activation, followed by an effector function. That means if the antigen is being recognized, there will be obviously an activation. When there is activation, it will lead to an effector function. This is just the scientific way of describing a simple process that the antigen is being recognized and there is an immune response. Recognition, supposing the antigen has gone and there is no response. We know that the antigen is there. That means it requires a co-stimulation. If there is a co-stimulation, then there will be energy. Energy is just the inability to respond. So, that means if I do not see a response, what do I understand? That there is energy, and energy is never into active cell. Energy is always on the peripheral cells, which I will describe later. What is activation? That means I have spoken about cytotoxic cells, T cells, I have talked about helper cells. Now, these are the cytotoxic T lymphocytes. That means once the T cells are activated, they mature into cytotoxic T lymphocytes, and this requires an CTL associated protein 4, which actually engages the cluster of differentiation 80 to 86. That means, what am I talking? I am talking about things which you don't understand, but it is basically the receptors, and this is going to cause the down regulation of T suppressor cells. Now, for 1 minute, just try to remember that if there is suppression, there is a reduction in the activation. So, that means my activation might be to increase or to reduce. Similarly, effector function also. That means if I go on giving an antigen, I might lead to causing a state when there is no response. When there is a no response, it almost becomes like an immune tolerance, or I can induce the state of unresponsiveness. So, when I talk of regulation by antigen, what do I understand? That I will induce the immune response, and this the immune response should also get extinct before after a point of time. What do I talk about? I talk about affinity maturation of B lymphocytes. I have to retain the immunological memory. I have to also remember that there will be antigenic competition. I will also remember that there is a threshold density. That means there is a certain level of immune response which is capable of making a complex and showing an MHC class 2 molecule. Otherwise, it will not express itself into the antigen presenting cell. That means it is not that if I am going to stuff an animal with antigen and expect it to get killed, no. The body has a system of regulating and surviving in the circumstances. So, what are the factors that are going to influence the outcome of the immune response? The same antigen can induce a very active response or the active state of tolerance. That means my antigen is capable of both, either creating an immunity or reducing the immunity. What all does it depend on? It depends on the state of the immune system, on the properties of the immune system, the properties of the antigen, on the dose of the antigen, as well as on the route of the antigen administration. That means my antigen, I had explained last time, body is most immunogenic to a protein as an antigen, slightly less lipids, and carbohydrates are least immunogenic. Second important point is the dose of antigen. The amount of antigen I given also I can infuse the animal with a lot of antigen, and it might cause an immune response much higher than the others. Route of administration is also important, because if I give a subdermal or an injection in the muscle, I can elicit a better immune response than when I inject it into as an intravenous. As an intravenous, the immune response is least. So, if I look at this, I've just made a small example of an experiment which had been done earlier. That means if I put a virus, I can see the antiviral toxicity. I can see the Th1 response. Th1 response is dependent on the levels of interferon gamma which we see, and Th2 response we see in the level of the interleukin 4. And what do we see? That there is a difference in the dose as well as route of the antigen exposure. So, is there a role of antigen receptors in the immune response? This is a question which can be asked. Yes, if we are understanding everything, that there is an antigen, there is a B cell, there is a T cell, there is activation, there is MHC molecule, there is antigen presenting cell, but we must also remember that there are receptors which are present on the cell surface which are responsible for recognition. So, will B cell and T cell both have the receptors? Yes, they both carry customized receptor molecules which will allow them to recognize and respond to specific antigens. The B cell's antigen specific receptor sits on the outer surface, and it is also prepared to make more antibody receptor antigens and create more and more space for more antibodies to be recognized. But the T cell receptors are not surface bound. They are more complex in nature, and they can only be recognized after the antigen has been processed. That means, as I had explained, it will only recognize the MHC molecule which is expressed on the cell surface of the antigen presenting cell. But natural killer cells, do they recognize the antigens as class 1 MHC markers? Yes, they do. And the helper cells will recognize the antigens only as class 2 markers, MHC markers. That means this entire thing also tells us that B cell and T cell both also have specificity. It is not that the natural killers are just natural killers. They are not just the background like I explained. They also have specificity, and the specificity is based on the expression of and recognition of MHC class 1. So, looking at the antigen receptors, I am again going to show you a cartoon which very categorically shows that the antigen of the B cell, the antigen receptor present on the B cell will recognize, and the antibody expressed on the cell surface are responsible for making a combination. So, that means there is recognition. If I talk of the killer cell, the natural killer cell, what do I see? There is again a complex which shows the T cell receptor, the antigenic peptide which is responsible for recognition, and you have the cell surface expressed MHC class 1. As a result, there is a combination which leads to specificity. I am explaining the specificity. As far as T helper cell is concerned, here what we see, a T helper cell will always recognize an MHC class 2 molecule. So, an MHC class 2 molecule which is expressed on the surface of the antigen presenting cell will be recognized by the T helper cell. T helper cell has what? It has CD4 receptor expressed on it. This makes a complex, hence the T cell complex is more complicated, but it makes a recognition which is highly specific. So, T cell, T helper cell and cytotoxic T cells, they do not go and bind non-specifically. They will bind to the MHC class 1 and class 2 molecules which are specific. Now we will take a break, and after that we will start the regulation of the immune response part 2. Good evening and welcome back. So, we proceed to the regulation of immune response part 2. Like I had been discussing that there has been regulation based on antigen. What was the regulation by antigen? There was recognition, there was activation, as well as there was effector function. Now, the effector function was basically to produce antibodies. The body has another regulation, that is, they will also regulate the amount of antibodies produced. So, what are the amount of antibodies which are produced? They will compete with the B cell receptor for antigen. Like I also said that there are antigen receptors which are present, which will lead to specificity. So, that means that now the antibodies which are being produced are either as soluble mediators in the blood, or they will bind to the cell surface receptor and they will compete for the same site. When they compete for the same site, there is a negative regulator of B lymphocyte, hence there will always be a balance. Antibody immune complexes will bind to the BCR and the FC receptor network on B cells. That means there should be, I'm sorry about the spelling, there are FC is the part of the immunoglobulin, which is the part which goes and binds to the antigen presenting cells. This is the part which is conserved. It is the other part, the Fab part, which has the ability to recognize all the antigens. So, the antigen binding sites are on the Fab portion and the FC portion is the conserved site which goes and binds to the macrophages. So, the complexes which will be formed are being recognized by the surface of the B cell, or they will be recognized by the macrophages. This in itself will stop the B lymphocytes in binding the antigen again and again. Also, it is still not very clear whether the regulation is via an idiotypic network. Now, it is important to understand what is an idiotypic network. When I talk of idiotypes, idiotypes are types which are recognized or they have the same type. That means if my B cell recognizes a certain type of an antibody, it will recognize the same antibody and that antibody will go and bind to the same place. So, this is an idiotypic phenomena. But this idiotypic phenomena is not very important as far as B cells are concerned, but it is very important as far as T cells are concerned. Looking at that, I will discuss with the T cells. Coming back to my activation of B cells, because this activation will lead to the making of antibodies. We remember we have all this cartoon will also explain exactly what I have explained earlier, that the B cell needs to get activated to give the antibodies. This is just a little revision so that we do not forget what we had started with. So, the surface of the antigen will recognize the antigen antibody complex and then present it to T cells. Further on, the B cell which will recognize the antigen, a part of the antigen, this is called as the epitope, and the part of the antibody which binds to the antigen is called as the paratope. This itself will lead to the formation or the expression of MHC class II molecule, or it can also lead to the production, clonal production of the antibodies itself. When I say regulation of antibody, I talk of idiotype, anti-idiotype. That means antibody interactions can stimulate or inhibit the antibody responses. That means here I have an antibody, the Y-shaped is the antibody. I have the antigen bound to the antibody, that means my Fab portion is completely saturated. This is the FC part which is bound to the cell surface, and this is leading to no activation because my antibody here is saturated by another, the antigen is saturated by another antibody, and here there is no regulation. So, if there is no recognition, this in itself is a regulation. So, that means an antibody is blocking an antibody complex leading to no response. When I talk of an activation and effector function in case of antibody, like we spoke about the antigen, what do we see here? That the FC receptor is there, the antigen is bound, again this antibody has no activation. So that means the Fc receptor, although it comes and binds to the cell surface, it leads to no activation. So there is a cross-linking of the receptor of the antigen antibody complexes. Although they bind to the Fc receptor, they do not send an activation signal, rather they send an inhibitory signal. So there can be another regulation by the antibody itself. That means an antibody-antigen immune complex which is bound to the complement system. Now, complement system is another system where the proteins of the serum or of the blood are capable of creating and killing the bacteria. That means they make an inhibitory complex which will kill the bacteria itself. It is a process which is a complete different chapter which will be discussed with you all later. So, here the antigen antibody immune complex may bind to a part of the complement system, and it might localize the impact of the antigen presenting cells through the complement system and may control the antigen. As a result, the antigen, the antibody and the complement system together can form a complex which can either lead to an activation which will show the complex as activating the cells rather than inhibiting. So when I talk of idiotypic regulation, what did I mean? Idiotypic just means a sum of all idiotypes. That means the variable determinants in a given antibody molecule of the T cell receptor. That is why I had said it is not as important in the B cell, but it is very important for the T cell activation. When I talk of number of idiotypes, it is very interesting that the idiotypes expressed on all the cells are called as public, and which are expressed on certain specific cells, I would call them as private idiotypes. That means the ones which are unique to a particular cell. So that means there is an idiotypic regulation of the immune system as well. When I say there is an anti-idiotypic, what is an idiotype? Which is present on all surface of the cells. And this are similar variants. That means all the variants expressed by different cells are same. So if I have an antibody which is against these idiotypes, I will call it an anti-idiotype. When I have this anti-idiotype antibodies, they block and regulate the immune response because they will be recognized by the antigen binding sites and they will block the antigen binding sites. As a result, they will create an immunoglobulin pool and participate in both humoral as well as cellular immune response. I hope it is clear. Anti-idiotypic antibodies. Idiotypic are the variants expressed by all the cells. So antibodies against these idiotypes will be called as anti-idiotypic antibodies. This idiotypic network recognition was recognized and given a Nobel Prize to Jern in 1984. What did he explain? He said that idiotypic determinants are in itself immunogenic. Anti-idiotypic antibodies are formed and they can actually suppress the antigen. Anti-idiotypic antibodies can induce an anti-idiotypic response. That means an response against the idiotype is anti-idiotypic. I can generate a response against the anti-idiotypic antibodies too. That means this will actually slowly fade away the response against a certain antigen. That means the gradual fading of the immune response becomes a reason for the immunological tolerance. So this entire network was explained by Jern, which got him a Nobel Prize. This is a very important part of understanding immune regulation. So what have we understood? That there is an idiotype, which is a variant expressed on the cells. There is an anti-idiotype, which is the antibody against these variants. And there can be anti-idiotypes, which are going to be the cause of the response fading and causing immune tolerance. Well, we have done role of antigen, we have also understood the regulation of antibodies. Now we have to understand the role of cytokines in immune response. We also must remember that these are small molecules which are like hormones, and they are secreted in the blood, but in very small quantities for very short periods of time, and these chemical messengers can elicit and communicate a lot of response from all the cells. That means the T cells, the antigen presenting cells, every cell is very sensitive to the stimulation of these small cytokines. These are just small peptides. Lymphocytes, that means both T cell and B cells, are capable of secreting cytokines, and they were called as lymphokines. Cytokine is another chapter which will be taken up in a detailed form much later, but just to have a little brief for understanding the importance in immune regulation, I will just mention a few types. There are interferons, which I have been talking about, interleukins, tumor necrosis factor, TGF, that is transforming growth factor beta, alpha, and there are certain small chemical messengers which are called as chemokines, and these chemokines are very important in inflammatory response to induce an anti-inflammatory cascade of increasing the recognition and recruiting of the cells. So, to understand the regulation by cytokines, that means my cytokines like interferon gamma, I had told you, was for Th1, and IL-2, that is interleukin 2 or 12, was for the Th2 response. So that means my cytokines are both as positive as well as negative regulators. That means they can act at different stages of the immune response. They also depend on the milieu where they are required. That means what is the condition of the body and what are the conditions of the cell, and how and when and how much are required at what point of time. Like I mentioned, the cytokines are extremely specific. They have the specificity, that means a small amount has to be highly effective, so they should have specific receptors for their recognition as well. So that means cytokines are capable of regulating the immune response, and they can determine the extent of the immune response to be generated. That means my cytokines can be a regulation on the cellular cells. That means it can interact with the antigen presenting cells and the T lymphocytes. It can interact with Th1, that is through the macrophages. It can interact with Th2, that is B lymphocytes. There can be otherwise also a control of Th1 versus Th2. That means T helper cell 1 will regulate Th2 response, and a T helper 2 response will regulate the Th1 response. We will come to that also. Also, it can have a complete regulation on the development of the leukocyte subpopulations. This is again important. That means whether I require more of neutrophils, I require more of acidophils, I require more of basophils. If there is an allergic response, definitely we need more of mast cells. So these kind of recognition and development can be stimulated by cytokines. When I talk of the negative regulation is cytotoxic T lymphocyte associated protein 4. This is an inhibitory receptor which is present on T cell. Like I said, this binds to the ligands C8 cluster differentiation 80 and CD86. I spoke of humoral immunity. I'm going to talk of cell-mediated immune responses. When I talk of cell-mediated immune responses, I'm giving you a cartoon of the role of cytokines in suppression of the cell-mediated immune responses. What do I see? I see an antigen presenting cell and I see a naive T cell. When I say naive T cell, that is a T cell which has not seen a complex or the MHC molecule of that particular antigen. That means it has not been subjected to the antigen which is present. Once it recognizes, there is secretion of IL-12. This naive cell will then activate the effector cell. Effector cell meaning that means that it is going to recruit T helper cell 1, which is going to start secreting interferon gamma. Now, once the interferon gamma has been secreted, the interferon gamma is a cytokine. This itself is going to cause activation of the macrophages. It also leads to T cell proliferation and differentiation. That means you require more T cells so that the recognized antigen through the antigen presenting cell may be killed. So, there has to be proliferation and differentiation of T cells leading to the effector function. Simultaneously, cytokines which are produced because our body can always not be in a state of activation. The body has to come back to a normal state. That is why a regulation of immune response is very important. I cannot allow the body to be stimulated in such a way that you are secreting cytokines all the time. Nature does not permit that. So, simultaneously the cytokines have to be countered by another set of cytokines which are called as cytokines which are produced by suppressor T cells. What are they going to produce? The suppressor T cells produce IL-10. IL-10 has the function of inhibiting antigen presenting cells. So, that means T suppressor cells in itself control the antigen presenting cell, antigen presenting cell controls the T cell controls the activated macrophages, but the inhibition is by TGF beta 2, that means transforming growth factor beta that inhibits the T cell proliferation. That means if too many T cells are being produced, proliferation just means increase in the population. If more T cells are being produced, TGF beta is secreted so that it stops the proliferation. If more antigen producing cells the antigen is being presented by too many antigen presenting cells, then IL-10 is secreted. IL-4 is secreted when there is too much interferon gamma in the system. So, that means excess interferon gamma can also put the body in an antiviral state, because body recognizes interferon as a secretion that the body is under a viral attack. So, the entire body is in an antiviral state. So, the moment there is interferon gamma, IL-4 is secreted, which is going to inhibit the secretion of interferon gamma. Similarly, IL-10 can also inactivate the macrophages. So, that means IL-10 controls the antigen presenting cells as well as it controls the macrophages. Interestingly, we have finished antigen, regulation of antibodies and regulation of cytokines. The next I am talking about regulation of the immune response through the genetic controls. Now, there can be some antibodies which are present in the mice which respond to some antigens. That means they are called as responders, and there are many people which are non-responders. This is an experiment done in rabbits. But do we not all know that some people are susceptible to infections and others are not? So, that means there are some who constitutively are responders. They secrete more of their genes are capable of secreting more of antibodies and good antibodies. That means the genetic types of certain types are capable of producing antibodies and they generally have a good immunological health. Others are non-responders, so they are susceptible to a lot of diseases. And how are these genetic factors determined? These genetic factors are determined through the MHC linked. MHC, if we have not forgotten, it is the major histocompatibility complex linked genes which control the infection. That means I have certain HLA. HLA is human leukocyte antigens which are expressed on the leukocytes. Like the RBCs express the blood we make the blood groups based on the polysaccharides present on the surface of the RBCs. HLA are human leukocyte antigens that are expressed on the surface of the leukocytes, and they are the ones which are responsible for making good responders and poor responders. That means susceptibility versus resistance. That means this leads to excess cytokine or chemokine production, maybe depending on the receptors. The non-MHC genes may also be capable. That means my macrophages can be regulated. The activity of the macrophages can be regulated through non-MHC factors. So, we have understood the humoral immunity and we also have understood the cell-mediated immunity. Interestingly, Th1, which is responsible for cell-mediated immunity, secretes interferon gamma and inhibits the Th2 response. That means excess antibody production. If there is excess antibody production, this will give out IL-4, IL-10, THF-beta, which is going to control the cell-mediated immunity. So, this shift of Th1 to Th2 is a paradigm which still needs to be worked out. Why exactly is one being controlled by the other and how important it is in the system. So, what have I understood? That there has to be a balance, a homeostasis of the immune system. There is an aggressive immune system of the responders. There are regulatory immune responses which are now controlled by regulatory T cells. T-regs or T regulatory cells are very important. Now, I will take you back to the cellular control, but quickly, that we have the T helper cells. We have 5 subsets: T helper, cytotoxic, then T suppressor, cytotoxic T lymphocytes, and we also have a T cell homeostasis, which is important, which is going to maintain how many T suppressor cells, when the T suppressor cells have to work, and how many T helper cells are going to activate the cytotoxic T cells. That means my T cell activation will have to determine the amount of immune response. That means T cell activation is going to lead to T cell expansion or proliferation. This might lead to anergy. I said anergy is an important part where you control the cells at the peripheral level, not where the things are happening. That means my activated T cells, if they are too many getting activated, they have to go under apoptosis and they will be killed, but only what will remain is the surviving memory cells. That means my memory cells were part of the B cells, not part of the T cells. So, this is just a response of the body after the T cell has been activated after a certain point of exposure. Now, the killer cells, suppressor cells, memory cells, helper cells, we have already gone through this. Why do I need an activation of T cells? That also we have understood, that the activation occurs through the T helper cells, MHC class 1, MHC class 2, through the cytokines or the lymphokines or the chemokines. As a result, we have all 4 kinds of cells activated, one which is doing the activation and the other which is doing the suppression. Now, this is helper cell, will recognize the antigen only on the surface of the white blood cells, on the macrophages. Again, activation of T helper cells, I will not go through the cartoon again. Killer cells, we have also called as the cytotoxic T cells. I told you the CTLs, that is cytotoxic T lymphocytes, they are different to T cells because they are the ones which are the effector cell of the activated T cells. T helper and TC cells do not kill, but they are killed by the actually the tumor cell will be killed by the cytotoxic T lymphocyte. Memory cells of the T cells are also as important as the memory cells of the B cells that produce the antibodies. The both of them can activate the killer cells. So, that means killer cells, cytotoxic Ts and the natural killer cells together form a complex which can kill the antigen, which can actively kill the antigen. So, that means I have a regulatory cell which is a suppressor cell, and there are 2 models of function of the regulatory T cells. One, that the T cell competes for the same antigen, and the other scenario is when the T cells compete for the cytokine signals. That means my cytotoxic T cell will activate, or they will be activated by the mature dendritic cell. The mature dendritic cell can either control it directly or it can get activated by the regulatory T cell. This regulatory T cell is as important as the plasma cell. When I have the cytokine, this thing again it is competing for the same sites. That means my regulation by T regulatory cells will not prevent the initial activation of T cells, but it will have an impact on the prolonged response of the system. That means any chronic disease or any chronic response will be inhibited by the T regulatory cells. So, T regulatory cells do not show a T1, Th2 or Th17 response, but it definitely suppresses the Th1 and Th2 responses. So, T regulatory cells are also arising very naturally. They are also arising through the thymus, and they are very important because they are responsible for FOXP3, CD4 plus CD25 response, which is actually called as the T regulatory response. This looks after the CD25 part of the IL-2R receptor. That means what is FOXP3? It is a transcription factor which is more in defect in the autoimmune disease or in the inflammatory disease. So that means what can my T regulatory cell do? My T regulatory cell can further suppress the cell in a cell dependent manner. This mechanism has to be worked on. So regulation is very important. Suppression by T regulatory cells is equally important. What is my conclusion? My conclusion is that I started with the first slide. My first slide said there is activation versus tolerance. The activation is the immunity the body has and tolerance. What have we understood till now? That the immune responses are the outcome of a balance between immunity and tolerance. The mechanism of unresponsiveness has to be worked out. That is worked out based on the antigen which is given, how the antigen is given, and the tolerance. This dogma of central tolerance depends on the deletion, it depends on the receptor, the editing, and the peripheral tolerance depends on the energy, that is the unresponsiveness of the cells. That means it can be clonal ignorance or suppression. Suppression occurs because of T regulatory cells. So we need to remember that humoral immunity, autoimmunity, and T regulation, regulatory cells are equally important. So this is a cartoon again which I just wanted to show. This is the last slide, the central, that is clonal depletion receptor editing, the clonal deletion receptor editing, that is from the peripheral cells, energy which leads to T regulation and suppression.
Okay. On that note, I would like to thank ma'am for this very enriching discussion, and I would like to thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.
Thank you.